Okay, so welcome back to this next video in which we are discussing the involvement of glycine receptors in progressive encephalomyelitis with rigidity and myoclonus. Okay, so we are discussing the structure of the glycine receptors within the spinal cord. We've seen their role in the inhibitory neurotransmission onto the alpha motor neurons, which innervate the skeletal muscle cells. And now we're going to look at their structure. So, I told you in the previous video that there is not just one gene that codes for the glycine receptor subunit. Instead, there are 5 genes which encode for the glycine receptor subunit. So you have 5 different subunits for the glycine receptor, okay? And these are grouped into, well, they're grouped into 2 families, I suppose. There's the alpha family, which contains 5 genes, the alpha 1, the alpha 2, the alpha 3, and the alpha 4. So those are 4 genes which all have slightly different sequences of amino acids, which leads to their protein, they all have slightly different sequences of organic bases, which leads to their proteins having slightly different sequences of amino acids. So they're all slightly different proteins that you produce from those genes. However, they're similar enough that they will all have the same membrane-spanning topology here, and they can all be used as a fifth of a glycine receptor. Then there's one final gene, which is known as the beta gene. So, we have these 4 genes here, the alpha 1, alpha 2, alpha 3, alpha 4, and then we have this 5th one here, the beta gene. And they code for the alpha 1 subunit, the alpha 2 subunit, the alpha 3 subunit, the alpha 4 subunit, and the beta subunit. Now, how do you actually compose these together to make a glycine receptor? Well, the reality is that there is actually only one glycine receptor that we need to know about, because it's the one that's important postsynaptically. It's the main one that's important in the body. Okay? And this is what's known as the alpha 1 beta heteropentamer. Okay? So, hetero means different. Pentamer means that it's composed of 5 subunits. So basically what this means is that when making one of these glycine receptors, you will use 2 different types of subunit. You will use the alpha 1 and the beta types of this subunit. So you won't ignore the other 3, alpha 2, alpha 3, alpha 4, scratch those off. These are the 2 important ones, alpha 1 and beta. Okay, now I would love to be able to tell you the exact structure of this alpha 1 beta heteropentamer, but it's not clear basically. We're not totally clear on the stoichiometry even of the receptor subunits, and it may well be that the glycine receptors do not have a fixed stoichiometry of this, that there are different stoichiometries used. Indeed, that does seem to be what's emerging. So we think that the stoichiometry there may well be 2 main stoichiometries, which is 2 alpha 1 subunits and 3 beta subunits, or 3 alpha 1 subunits and 2 beta subunits, i.e. you'd make 2 alpha 1 subunits and put them together with 3 beta subunits, or you can make 3 alpha 1 subunits and put them together to make 2 beta subunits. Both of those seem viable options for an alpha 1 beta heteropentamer. So what I can tell you is that there are these glycine receptors. The main form of glycine receptor that is found in the spinal cord and is important in glycinergic neurotransmission consists of both alpha 1 and beta subunits, okay? Now, we do not know for sure what the stoichiometry of these receptors is, and we don't know either the position of the subunits relative to one another. So let's say it was this stoichiometry, we certainly don't know how they're positioned relative to each other. So for instance, if this is the receptor viewed from above here, with the pore down the middle, we don't know whether, you put the 2 alpha 1 subunits next to each other, like so, and then the 3 beta subunits next to each other, or whether you put some beta subunits in between the 2 alpha 1 subunits. We don't know how they're oriented relative to one another. So this information just isn't known. It's not a widely studied receptor, not compared to the nicotinic acetylcholine receptor or the GABA-A receptor. Okay? What we do know is that the glycine receptors that you find in the spinal cord, the main type, consists of both alpha 1 and beta subunits, and that it's gated by glycine. We also think that the glycine binds to the alpha-1 subunits of this receptor. But again, since we don't know how many alpha-1 subunits you have for certain, we don't know how many glycines are going to bind either. But we do think that they bind to the alpha-1 subunits, and that's actually going to be important when we discuss progressive encephalomyelitis with rigidity and myoclonus. Okay, right. So, that now is all I have to say about the glycine receptor. So let's now talk about progressive encephalomyelitis with rigidity and myoclonus. Okay, right. So what happens in progressive encephalomyelitis with rigidity and myoclonus? Well, basically, first thing I should say is that there are different forms of this disease, basically. So there are different things that can be targeted in different forms of PERM. We are going to talk about the role of the glycine receptor in progressive encephalomyelitis with rigidity and myoclonus. So, basically what you find in people with this disease is that if you take a sample from their cerebrospinal fluid, which is the fluid which, remember, bathes the spinal cord and the brain and runs in that central canal down the middle of the spinal cord, if you take a sample of their cerebrospinal fluid, let's say through a lumbar puncture, what you find is that the lymphocyte level in the cerebrospinal fluid is massive. Okay, so you have around 60 times 10 to the 9 lymphocytes in a litre of cerebrospinal fluid. That is not good. Okay, so that suggests that there is some sort of, if you had a lymphocyte level of that, we'd think you had some sort of brain infection or something along those lines, or infection of the cerebrospinal fluid, okay, per litre. Right, now, what are these lymphocytes actually doing there? Well, basically, PERM is caused by an autoimmune reaction, and we'll continue this discussion in the next video.